the presentation. Otherwise, I want to acknowledge all of you. Kenneth Mobobia, a senior comrade from Meru, I can see you. Welcome. And we know you can confirm once again that you can hear me and maybe unmute and say hi.
Yes, Fred, I can hear you.
Very well. So I think you can proceed. You can proceed.
Okay, thank you. Good evening, everyone. Today we'll be talking about laboratory biosecurity, protecting science from misuse. So biosecurity is very key. It captures how we are going to protect the information that we handle in the lab and the biological material, that is the microbial materials that we handle. And so there are critical issues in the lab that really need to be guided. So for example, the biological material, if they are left in wrong hands, they can be used as bioweapon, and that could lead to a lot of biological threat or even a rising pandemic. So this is a good topic, and it is a gray area. So with the rising of ethical concerns and litigations in our country, and again, the advance in technology, it is also key that we consider biosafety and biosecurity, because the two goes in hand. So otherwise, I'm Francis Awino, a medical laboratory officer at the Nairobi Hospital, and I welcome you for today's webinar. Frederick, just confirm if I'm audible so that we start. So as we have said, the laboratory biosecurity refers to institutional and personnel security measures designed to prevent the loss, theft, misuse, diversion, or intentional release of biological agents being handled in the laboratory. So in biosecurity, we are looking at what control measures are there in place. And that's why even in the standards, ISO 15189 requires that there is a control access to the examination areas. And that one is very key because that control access is meant to protect the confidentiality of the critical information that we handle. That is one. The equipment that we are using at the lab. That is two. And then third, again, the microorganisms, or rather the biological materials that we are using. So it is very key. So those institutional measures, as an institution, you will come up with the measures and mechanisms on how to achieve this, and also the personnel security. You have to protect both the patients and as well as the laboratory staffs, or rather the clinical staffs working in a particular hospital. So those are measures that will be designed by a particular institution. So it will be institution based depending on the infrastructure that they have in place. And the key intention at the end of the day is to prevent loss, theft, or even attacks, or rather terrorism, things like that. So what are some of the challenges, especially when you are implementing biosecurity policies and procedures, which are aimed to control measures on a biological agent? One, there are many biological agents that can be found in their natural environment. And then second is biological agents of interest may be used in small quantity or may be capable of self-replicating, making them difficult to either detect or reliably quantify. It might be difficult quantifying them. And in some cases, the process of quantification itself may pose biosafety and biosecurity risk. Like cases where you are dealing with HBV, hepatitis B virus, for example. So even when we are trying to quantify, remember those are potential infections. And so therefore you have to have some engineering tools that will protect the personnel working around working around with such particular organism. So the process of handling or other manipulation, or when you are trying to quantify them, quantifying these particular microbial agents by itself or by default could cause biosafety and biosecurity risk. So that's also a challenge. So for this reason, biosecurity risk assessment and establishment of appropriate risk control measures is required. So before you can come up with biosecurity control measures, we need to do a risk assessment, and we'll see how we are going to do the risk assessment to come up with this, some of the control measures that you are going to put in place as a lab or as an hospital. So the risk control measures will be required and should comply with the national standards and the regulatory procedures. So there is a WHO guideline on laboratory biosecurity, and it entails what should be done, and it will give you a guidance on how you're supposed to do biosecurity risk assessment. And that will scale down. WHO is at the international level. So we as Kenya will come up with the national policy guidelines. And then now from there, the hospital base. So the hospital will also come up with their policy, or rather construct their policy based on the national guidelines, which is by the MOH. And I want to believe those policies are there at the MOH. So when you are doing biosecurity risk assessment, what are you going to do? So the first step, you will gather information. So the information that you are going to gather, one is the type of biological agents available and their physical location. So what is the biological agents that you are dealing with? Is it hepatitis B virus, for example? What is the physical location? So these samples that you are having, what is the potential pathogens that they are having, and how are you handling them? And who get access to this particular pathogen? Let's say it is a molecular lab, for example, where you are dealing with the DNA material. So how do you handle the DNA materials after you've extracted them? Remember, these DNA materials of these particular pathogens, let's say it is an HBV, they can be manipulated, or rather somebody can do some genetic modification which could rise into a bioterrorism, something of like that, or rather use them in the wrong way. So that's why we are saying in the biosecurity, you are protecting misuse of science, because one, you don't want you don't want your biological material to land in the wrong hand. And again, so the key information there, you have to understand the nature of the biological agents that you are handling at the laboratory level and their physical location. So the personnel required to access the laboratory premises, either to handle the agents or for the reasons, and those responsible for the biological agent. So which means that we must have the information of every single person that is coming to the lab, and also those ones who are working. So you need to have control of access, in that you are having a records of people who comes in and people who comes out. And so therefore you could use whichever method you are using in your system. It could be automated system where they are using their fingerprints, or rather biometrics, to access the laboratory. But again, you need to have a record of the activities that are going on and the personnel who are accessing this particular material in the lab. And then once you've gathered enough information, the next step will be to evaluate the risk. So the risk, how do you evaluate that? The likelihood of someone gaining access to the identified biological agents. What is the likelihood of someone gaining access to, for example, in your DNA bank, and not DNA bank, in the freezer where you are storing your DNA material, let's say for COVID, or rather COVID materials, or even the ones that have been extracted, or even the remnants or the amplicons. How do you dispose them? And how do you handle that? So those are some of the questions that you need to ask yourself when you're evaluating the risk, and the position where it is placed. How likely is it that unauthorized access can be granted, which means anyone can come and get access to this particular material. So likelihood of someone getting access to the identified biological agent. So first you have to identify the biological agents that you are dealing with, and then now you assess the location and where you place them. And again, the control measures that you are going to put in place to minimize access to these particular agents. And again, after looking at the likelihood, you will also look at the consequence of a deliberate release of those agents. So you ask yourself, if at all this particular agent is released, then what will be the impact to both the environment and the public? And then compare the two factors to establish what the overall and initial risks are. So you look at the likelihood, and then you look at the impact, that is the consequences in case of in the unfortunate events that these particular biological agents are released. And then now you look with that, now you can establish the initial risk stroke the overall risk. So the third step, once you've gathered your information and you've evaluated the risk, you will develop a risk control strategy, which in this case, you're looking at the acceptable risk, that is the minimum security standard required for work with the identified biological agent. So you've already evaluated your risk. And then now you look at what is the acceptable risk, the minimum security standards which is required to work with this particular agent. And then once you've looked at that, the next step is to select and implement the risk control measures. So based on your assessment, based on the information that you've gathered, and based on the evaluation report that you have, you will do assessment of the suitability of personnel. You will look at the personnel who are handling these biological agents. Do they have the prerequisite training? And they have also been trained in the biosafety as well as the biosecurity, because we are not going to implement without training the personnel. So assessment of the suitability of personnel, and then security specific trainings and rigorous adherence to pathogen protection procedures. So again, it is capacity building where you train your staff on how to handle those particular agents, or rather biological agents that they are dealing with. And here we are using microbial agents as an example. It could also be in terms of equipment. So again, you need to train your staffs on equipment and how they should maintain those equipment and the safety mechanisms when they are using those particular equipment. And then review risk and risk control measures. So the laboratory biosecurity protocol should be able to identify. So the protocols that we have in place should be able to identify, should be able to allow reporting, so should be able to report, should be able to investigate, and then remedy breaches in laboratory biosecurity. In case there's a breach in biosecurity, or in case there's unauthorized access to, let's say, a lab. Let's say, for example, you are working in lab with different sections, and there's unauthorized access to chemistry, or there's unauthorized access to your cold room where you store your blood products. So what is the corrective action in such a case? So you have to come up with a root cause analysis in the unfortunate events, or in case of any incident, and then you should be able to identify some of the remedies and how you can prevent the occurrence of the same. And so with the incidents that you are receiving, or even the complaints that you receive, that's how you'll be able to review the system to see whether it is functioning effectively or not. And based on that, you are able to modify your SOP, or rather amend your SOP. And not just the SOP alone, you will also be able to improve the system further by implementing control measures that works for you. So the next one, we'll look at the inventory control. So what is inventory control? It's a comprehensive program of accountability that is necessary to establish adequate control of at-risk biological agent and to discourage theft or misuse. So this can be achieved by compilation of a detailed inventory. And so your inventory should include this description of the biological agent, its quantities, storage location, use, the person responsible, documentation of internal and external transfer, and an inactivation or disposal of the material. So it is very important. Inventory system, if you have a proper inventory system, you will be able, this one will cut across. Biosecurity does not just restrict itself to the microbial agents alone. I said even the equipment, the reagents that are used in the lab, the safety of the personnel, all those. So it is very good, it is very key that you have a proper inventory management system. But if it is a biological agent, you have the description of that biological agent you are dealing with. It could be an HIV sample. So what is the quantities? How many have you stored, let's say at -90, for those ones who are working in research setup, or those ones which are due for sequencing? So how many do you have at your disposal? Where have they been stored? Maybe in freezer X. Under what compartment in that freezer X? And then the use, and then the person who is responsible for that storage, or rather inventory, keeping the inventory record and updating the inventory records appropriately. What is documentation of in case you've transferred this particular material to another place? It could be internally or externally. So if it is internally within the organization, again, you keep a clear record of that. Even if it is equipment, you have to fill the asset transfer form to the department that it is going for. The same applies to the external transfer. So that traceability in the system, that is all about biosecurity. And then inactivation or in case you no longer need let me use an equipment for this case. In case you no longer need that material or this particular equipment, let's say it is a chemistry equipment. So do you have a way of disposing? Is there do you have a way of disposing? So you have to fill in the form where it is going and how it is going to be disposed. So the disposal of the material or even inactivation, which means you are putting it out of service. So all those procedures has to be in place. And your inventory should be up to date. And so you should always review your inventory from time to time to ensure that the inventory management is up to date and that the right quantity, if your inventory system says you are having 20 samples, that one should tally with the physical count. Not that the system is saying you are having 20, but the physical count is saying 15. So how are you going to account for the missing 5? And the same applies to even your reagent that you are using. So it should be complete, accurate, and updated regularly to ensure that there is appropriate control and accountability. So after looking at the inventory system, we'll also look at the information control, because in the lab we generate a lot of information, and if this information is not properly guided, they could land in the wrong hand. And that's why you always you always hold your results with a lot of confidentiality, because most of the things you are handling there, you are dealing with you are dealing with the people's results. So you can imagine if a patient may be tested in lab A, and then they are not getting their results from lab A. Before even that results lands to them, they are already getting the information from somewhere else. So you could be put in a lot of litigation. So the information control has to be, your data protection and data management has to be up to date. And so therefore, biosecurity program must also have the processes and procedures used to protect the confidentiality and integrity of sensitive information. And that's why those LIMS, or rather the laboratory information system that you are using, their logins, and people are able to use login with their credentials, so that again you are able to keep the traceability. If maybe there is a modification or rather a change of information in the system, if an audit is done, you are able to tell who was there and at what time and what happens. And so therefore, the next one is sensitive information should be identified, labeled, and protected against unauthorized access. So the sensitive information, those ones that you don't want to land in the wrong hand, they should be password protected if that is possible, but that one should be identified. They should be labeled, they should be protected against unauthorized access. And so as an institution, we are going to come up with a control measures that will ensure that, or rather a system that will able to achieve this. The next sensitive information include research data. For example, in research data, most of those data, or rather the samples that you are using, they will be masked, or rather they'll be given unique codes. But remember, it is the principal investigator who is mandated to protect that information with a lot of confidentiality. And so therefore it will be the responsibility of the principal investigator to ensure that the data they are handling at the research is kept as anonymous as possible. So even the people handling the data, analyzing data, they don't know whose data they are dealing with. But again, if there is something they need, if at all there is a finding that they need to trace to a particular participant, then now the principal investigator will have the real identity. So that's why we say the sensitive information, they should be identified, they should be labeled and protected against unauthorized access, because the principal investigator here will have the records of the sensitive information, and they should be able to know that if it is code AZ will match person X or other person Z. And so therefore, diagnostic results, the one we deal with in the lab, those are very sensitive information and they should not land. So you cannot print patients' results and then just leave them flying all over. There are likelihood that they can even land in the hand of another patient. So you really need to protect the patient data as much as possible. If at all they were printed and they were not given out, you need to shred those particular material that are containing sensitive information of the of the patient. And then information on animal experiments. The same applies to animal animal science, where they're using animal for experiment. And then list of key personnel, for example, the IT people who maintain this particular system we are using, and biosafety contacts. Security plans, the access codes, passwords. The passwords should be kept as secret as possible so that you don't have unauthorized access to your system. The storage locations and biological agent inventories. So if you are dealing, most of us are in routine labs, but if you are working in sensitive labs like in Cambridge or most of the molecular setups, then you really have to have a proper inventory system of the biological agents that you are dealing with. So the next one, we'll talk about personnel control, because people sit at the center of the system. Without people, there will be no system. Because again, it's the people who interact with the system, and we are the one who carry activities. And then, so it is essential for the functioning of a laboratory, as it ensures that the daily work, practices, and procedures are being performed by suitable personnel who behave in a reliable and trustworthy manner. And so those two are the key. Are they reliable? Are they trustworthy? And so therefore, that's why prior to you being employed as a medical laboratory officer, you were in school for 4 or 5 years, or whichever period that you went through training, and you are told what is expected of you. That those ethics that guide every profession. And so that is how you will learn how important to keep confidentiality of the patients that you are handling, the type of data that you are handling, and all that. And so therefore, in recruiting, you must ensure that you are recruiting people of the right qualification. And again, you are doing their competency assessment to see whether the personnel that you are having are complying to both, not just the biosecurity and the biosafety guidelines, but to all the tasks assigned, using the various standard operating procedures guiding their practice, and also the regulation standards that are stipulated to guide the practice of the profession. So they must work in a reliable and a trustworthy manner. And so therefore, biosecurity procedures for visitors also and other outside personnel must be established to ensure that there's a legitimate need for access. So you have to come with the control measures on how visitors, or rather people who are not working in that particular area, or people who are not trained in that particular area, if they want to access lab, for example, where should they go to first? And that one should be kept. And the record of the visitors that comes in should also be kept. The designated areas that they are supposed to reach, at what point. That's why most of the doors, either if you don't have the biometric, then they should have the signage, authorized staffs only, so that you are able to control that particular access. And if at all they are going to access the sensitive areas like the testing area, then what other precautions should be put in place? And also, more important about the biosafety, you also look at the PPEs that they need to don before they can access those particular areas. So the laboratory biosecurity training for all personnel should be provided according to the outcome of the risk assessment. So we had already seen the risk assessment. And when you do your risk assessment, you'll be able to identify gaps in the system. And then now the management should be able to come up with the corrective actions to close some of those gaps. And so therefore, you don't just close gaps and keep quiet. You need to train your following audits, or rather risk assessment. You need to train your personnel on areas that have been identified as gaps. If, for example, there was a breach in confidentiality, then we need a retraining in that area. If, for example, there is a breach in access in the lab, then you need a retraining, and you will be emphasizing on the importance of keeping strict control access to the testing areas as well as the laboratory areas. Equipment, all the same. So you really need a lot of training for the personnel, so that you make them understand the reason as to why we are implementing some of the biosecurity measures in the lab. And then training to focus on need to protect biological agents, the national standards, and institution-specific measures. So as I said, some labs are dealing with very sensitive and highly infectious agents. It could even be Ebola, it could even be hemorrhagic viruses. And so therefore, you really need to train the personnel who are working in that particular area. Could be people who are working in molecular setup. They should be trained on how to carry out themselves in that setup, because it will not be the same as the routine lab, so that you protect your staff against the infectious agents that you're handling. Again, not just them, you also protect the release of these particular agents into the public. And so therefore you look at the storage of those particular agents they will be handling, you train them on that area. And again, you make them understand the impact in case of the unfortunate events. So next we look at the physical security control. So these are measures which will be put in place to prevent unauthorized access of outside adversaries, and also to minimize the threat from insiders, and promotes both biosecurity and biosafety objectives by limiting access to the laboratory and other potentially hazardous areas. For example, you cannot give access to everyone to microbiology example where you are doing your cultures. So that's why you need that physical security control. To clinical chemistry lab or even, so in the lab, you must put the physical security control measures. And not just in the lab, in the hospital also. And that's why most hospitals, you walk to any, most of these major hospitals, it might not be every hospital, major hospital, even at the gate, there's the screening by the security. So that is one of the physical security control measures for that particular institutions. Let's not just look at the lab alone. So again, they are also preventing unauthorized. For example, somebody walked in with a bomb, and that's why you have to go through that machine so that they are able to screen at the gate. So you are minimizing unauthorized access, and you are protecting your entire hospital community. So these elements will include boundaries. It will include access controls, like you've seen in most labs, authorized staffs only, or no entry beyond this point. Intrusion detections, that those alarms, in case of unauthorized access, they just the alarm system will alert the team who are monitoring the physical security control. So there will be alarm assessment and response to alarm. And this is very important, like even when we are doing the fire drill. So once the alarm is activated, everyone knows what to do. So before they know what to do, it means there's a training that has taken place. So it is also important to train your staff on how to respond to various alarm systems that have been installed in the institution. So the next one is transport control. So in transport control, you must comply with the national, international rules for packaging, marking, labeling, and documentation. So because we are working in the lab, we have most of the time we could be sending our samples either to national referral labs or even abroad or international, or even abroad. So if it is international, then you follow the international rules for packaging. Even for the national referrals, you have to transport them in the right temperature, the right material, and all that. And you also must keep the records of the people who are involved in the transport chain. That's why they need to have those shipment logs. It has to be documented from where it is coming from. I've released 2 samples sent by who, let's say person X, it's the person receiving who's transporting them, courier is who, and then when they have reached the other side, they have given that sample to who, and then who has received it on the other end. And you see, so that one will help traceability in case there is a loss or there is a theft, or in case there's a loss or a theft, then you will be able to track. And then again, packaging is very important to preserve the integrity of the sample that you are sending out for analysis. So that is very key, and you must ensure that your sample is in the right, again, also to prevent leakage. Yes, and unnecessary. Again, if there's a spill, then it should have something that can contain that particular it so that you don't end up exposing the people who are handling those particular samples during transport. They could not be medical personnel, and so therefore that is very key. But before they take that job again, they are also trained on the kind of material they will be handling and how to go about it. So in case there's a transfer, this one should be pre-arranged and pre-approved by responsible parties. And this can use chain of custody documentation based on the outcomes of a biosecurity risk assessment. And the inventories must be updated, as we have said, to reflect the incoming and outgoing specimen. So in case there's an emergency or rather incident response. So effective incident response is a risk control strategy, that can reduce the consequences of these unknown events through planning and preparation of potential incidents. And so that's why we normally do even spill drill, just to see whether the system that you've put in place is functioning effectively. And through those drills, you are able to identify the weak areas. So you don't wait until the situations, or rather the incident occurs, is when you test your system. So sometimes it's just good to provoke the system. The fire drill is necessary, just to provoke the system to see how people respond. And then from there, you have got observers, people who are able to tell you that this is where it was done well, or this part was not handled well, and these are the areas of improvement. And so looking at that, and we can look back and correct and even come up with a stronger system than the one that we had initially implemented in our facility. So an incident response protocol should be written and followed to ensure proper reporting. So we should have a clear guideline on how, in case of an incident, what happens. And let's say this, the most junior staff have encountered incident, they respond to them. So there should be that chain of response, and how escalation of events takes place in unfortunate cases. And then this one will facilitate investigation, root cause analysis, corrective action, and process improvement in general. So drills and exercise can be used in the planning and preparation stages to test the response to simulated incidents or emergencies. As we have said, major disaster response, you just do a drill on major disaster response. If you want to see how our doctors will react in case of a major disaster response. So before you do that, you have to train the people who are in the system. And each and every department must understand their role in major disaster response, for example. So that is why the drills are necessary. And so therefore, when we do these particular drills, then we are able to identify the gaps in the system and come up with corrective actions, so that in case of a real scenario, then we'll be able to capture events, and you'll be able to go in a long way to prevent harm or even death. So they can help identify gaps and other improvement opportunities, as we have expounded. And then plans should be reviewed and updated at least annually. And the information obtained through drills, incident reports, and investigation should be used to make necessary adjustment and improvement. So the senior management team in that organization will be able to sit and review some of the drills report that they have carried out, and then what were the areas of improvement. And then based on that, you can also be able to identify some of the weakest links in the policies and the guidelines that are in place, so that amendment can be done. And then amendment, and when you make an amendment to such, you also have to bring it to attention of everyone, which means you don't just amend and keep the paper. No. Once amendment has been done, it is something that has to be implemented across the organization. And how are you going to do that? You do sensitization of every personnel that is involved. So we have got emerging biological risk. So this one we look at, they include genetically modified microorganism, synthetic biology, gain of function research, stem cell research, gene editing. And so there's a lot of emerging biological risk, because we could be doing a study and there's genetically modification of microorganism. The organism could be non-pathological. Some people could genetically modify them to be pathological in such a case. And so that is a biological risk. So synthetic biology, gain of functional research, stem cell research, gene editing, and gene drives. You can edit a gene and make it lethal when actually in its natural or the original state it was not. Consideration on the risk posed by the incidents or the potential deliberate misuse of life sciences research should be put in place. And that's why before you take any research, you have to go through the ethical review committee, because that's where you are going to tell them how you will ensure that the biosafety and the biosecurity is properly implemented, from how you are going to protect the data, how you are going to maintain the confidentiality of this particular patient, how you will be handling your waste, how you will be handling your patients, or rather your participants. We are so used to the word participant, but in this context, I'm now talking of research. How you'll be handling your participants, and again, how they are going to benefit out of it. So it has to be ethically approved. Yes. So once you've demonstrated beyond reasonable doubt, and remember, this one doesn't stop at the point of seeking for approval alone. They always review, even in the past, when you're doing your now collection of data, going on with your scientific research and all that, they will always review the activities you are carrying out just to see whether you are really keeping the ethical guidelines that you are given before you start. And so now you will seek approvals from the organization who will be doing the research all the way to the national level. NACOSTI has to certify, and then now you can go ahead with that particular research. So those ones are very key, very, very, very key. And select appropriate risk control measures to mean, again, if it is something that is going to put a threat to the public, then it will not be granted. And that's why that ethical approval is very important. And that's why the ethical review committee is there just to ensure the safety of the public and the study participants. So you'll select appropriate risk control measures to minimize those risks in order to conduct necessary and beneficial life sciences. Remember, it should be at the maximum benefit of the society. As much as you want to generate knowledge or get a new finding in a particular area so that you are going to improve. So they will weigh the two between taking the biological risk and the outcome of the research. So the benefit must outweigh the risk. If the risk outweighs the benefits, then that research is invalid. So conducting research with emerging technologies for which limited information exists, the scientific community must, one, promote a culture of integrity and excellence. So we have to be people of utmost integrity in what we do. You have to be honest with yourself and with your findings. Distinguished by openness. There is no opaqueness in the system. You have to ensure that whatever you are doing, it is open and it is something that can be audited, and it's something that can be followed, and something that can be assessed. Honesty is very key. Accountability is very key. And that's why you have to do proper documentation for accountability and responsibility. There is no blame game when you are doing biosecurity and implementing biosecurity. It has to be who is responsible for what and at what level. So that one has to be clear in the entire organization. That's how you are going to come up with a proper responsibility. And you must create a blame-free culture within the organization. An organization where people can even self-initiate incident investigations. And then themselves, they can come up with some of the corrective actions or even the root cause analysis to search some of the incidents. And then that way you or everyone in the system, the people will remember we talk about the hierarchy, the level of seniority. It will be discussed at different levels until at the lab level, which is an hospital, will be able to review your SOP. Then at the hospital levels, they will be able now to review the policy. Because again, the policy will be reviewed at the hospital level. Again, now the hospital level, you've been moved to the national level now, or rather the county level, or at the regional level. So at that point, again, they will review the policy. Then when we reach the tail end at the national, we'll be able to come up with a guideline that will shape that particular practice or rather framework. So provide direction for biosafety, biosecurity oversight, and the risk assessment process for emerging technologies in life sciences. And finally, monitor and assess the scientific, the ethical, and social implication of certain biotechnologies. So dual use of research concern, this is life science research that based on current understanding and has the potential to provide knowledge, information, products, or technologies that could be directly misapplied to create a significant threat with potential consequences to public health and safety, agricultural species and other plants, animals, and the environment. So awareness of the dual use of agents, equipment and technology should also be considered in the development of laboratory biosecurity programs, national guidelines. And the potential misuse of biosciences is a global threat that requires a balanced approach to laboratory biosecurity so that legitimate access to important research and clinical materials is preserved. So as we have said, biosecurity is there to ensure that we are not misusing the bioscience. So we have to protect the access of the materials that we are dealing with, the information that we are dealing with. And we must train our people to understand what kind of information they are generating, or rather, what kind of data they are handling each and every single day. The biological material at their risk and what is the risk that it poses. So thank you so much. And that's all about biosecurity. I welcome questions at this point. Back to you, Fred.
Thank you, Francis, for the very wonderful presentation. And I think it's been very insightful, and this is a very, very good area that I feel you have handled very well. And maybe at this point, I want to invite any comments or any questions. You can either raise your hand, I'll give you a chance to speak, or also you can just give us your thoughts in the chat box. Otherwise, I want to appreciate your comments. They've been very kind. Keeping it, Samuel saying, this was very educative. Thanks a lot, Francis. Faiz Ndolo is saying, okay. And I think many of you who have been able to respond through the chat box. So allow me to invite any questions or concerns, if any, then maybe we can be able to discuss. This is an open discussion, an open forum, and you are invited to give your comments. Okay, so I was giving a chance to us. Maybe we can maybe just read. I think the presentation was quite clear. Dorothy Tinga saying, thank you for the presentation. Tunja Ruben, well elaborated. Josh Kirwa, well done. And Dorothy Tinga, Meshak Kenyuandege saying a good one. Joseph Chalo, educative and informative. Massimo Saru, very relevant presentation. Okay, so those of you who are asking for the slides, we can always share. So just reach out. So is there maybe anyone who wants to comment or maybe ask a question, or even just you have something that you want to share with us? You are free. You can either raise your hand or you can just share in the chat box. Okay, so meanwhile, maybe I just want to appreciate all of you. This is Corazon Akinyi, and she's asking, in case a fire drill has not been performed, what would you advise, and what are the other ways of auditing in case a plan is underway but not reviewed? Back to you.
So if a fire drill has not been done, then one, the organization has to review their policy. Because fire drill will be stipulated based on an organization plan. They could do it by annually or annually. So if it is annually, then they need to review their policy to capture that. But of course, the fact that it was not done, it means that that is already an unconformity to a particular policy. So if the policy is in place and it was not done, then a root cause analysis should commence, and then we address the reasons as to why the fire drill was not done. Maybe there was no planning. So if there was no planning for the same, a planning has to be taken. And let's say if it was for the year 2025 that was missed, it is good to do a mock drill within the organization. They can do a mock drill and then invite a few observers, that is a few observers, let's say from the people working with the firefighting agencies, and just invite a few to kind of examine their system, and we'll give them some of the gaps. And then once the gaps identified, let the organization work towards closing those particular gaps. And then once these gaps have been closed, let the organization now plan for a proper fire drill, for example. Yeah, so drills, as we had said, they are very important because that's how we will know how effective the systems that we put in place works. Yes, so the next one was?
Thank you. That's quite elaborate. I think there's another one from Simon Diwa, and is asking, in line with biosafety, biosecurity, do we have a criteria in which staff are vetted, trained, and authorized before being granted access to high-risk samples?
You are really breaking, Fred, but there's someone. Someone is
okay,
saying,
sorry for that. You can hear me now?
Yeah. Yes, I can.
Yeah, so I think there's a question from Simon Diwa, who is asking, in line with biosafety, biosecurity, do you have a criteria in which staff are vetted, trained, and authorized before being granted access to high-risk samples?
And that's why we said in our procedure, if you are not all the labs deal with the same potential threat, but of course every single sample is a biological. Every single biological material has got a potential threat if not handled properly, and so therefore we need to train staff based on needs. You look at the areas where they are deployed. For example, a staff that is deployed in the microbiology, for example, remember these are people we are culturing. They are dealing with the culture, and these are live microorganisms, and some of them are highly infectious. Even let's say like mycology, where you are dealing with fungal culture, those are highly infectious. And now biosafety, I will combine it with the question that another person was asking, how they can differentiate between biosafety and biosecurity. So in biosafety, we are preventing exposure to the infectious material, while in biosecurity, we are concerned with the access, unauthorized access to this particular potential infectious material. So again, now in the biosafety, we'll be looking at what are the engineering controls. Do we have a biosafety cabinet where they can handle this? And then after the work, where does it go? Now the biosecurity comes in. Is there an authorized access? Can someone come and access, for example, it was an aspergillus growing, and then they are able to explode this to the air, and so many people will be able to breathe and will be dealing with the more infection as such. So again, the training is very key. based on need and where the staff is placed. So first you do a risk assessment, and you have to know what is the residual risk and what is the overall risk. So I hope that one answered that question correctly. So the next one was?
Thank you. I think there's another one from Joseph Charlo. And he's saying, kindly answer this. How do we deal with biosecurity, biosafety in the lab to make sure the samples are secure?
So once we say you have to keep a proper inventory of your samples. Like for example, I don't know where he or she is working, but you could be, I'm just giving an example, maybe your system, you could be using a system like Labware, it could be Cranium, it could be Care. That's a different lean system that people use. But for example, Labware, for example, I've got an option of storing samples. So you are able to store your samples. And then of course you must have an SOP guiding on how you are going to store your samples. So again, you keep the inventory register. Like for example, sample A, it is stored in rack B. And in rack B, which compartment? Position 2. So even if you want to retrieve this particular samples, you can easily do through the system and also through the system is what will tell you where that track is in your storage room. So again, you have to come up with a policy. If it is a blood sample, you discard or either dispose after how long? Is it 7 days? Is it 14 days? And then again, how you discard again comes in. Where do you take your samples to? So that you don't end up posting. Now the biosecurity, the first one was biosafety. Now the biosecurity, you are now releasing it to the public. So if you are not doing the incineration yourself, now you have to ask yourself, you are having a contractor who is coming on site to collect these particular samples. As the organization vetted that particular contractor and ensure that they have got proper facility to handle the pathological waste so that we don't end up having pathological waste being disposed on landfills. So such key things are very, very important. So the next question.
Thank you, Francis. Thank you. I think we have handled, almost everything here. And Caleb, I'm sure you're asking for a presentation on biosafety also. I think we shall plan and bring that presentation. So, yeah, I think Joseph Charlo is saying this is well answered. Thank you, Joseph, from Peketoni. Thank you. Lamu. Simon Letting is asking, kindly, who exactly by training qualifies to do a risk assessment in the lab?
So the risk assessment in the lab, we have got the biosafety officer. So the biosafety officer is responsible for implementing both the biosecurity guidelines and the biosafety guidelines within a particular lab. But this one doesn't mean that the biosafety officer is the only one that is trained on the areas of biosafety. And that's why it is very important that as a lab or as an organization, we ensure that all our staff have received a training on biosafety and biosecurity. Because if you're just going to give them instruction without training them, they'll not be able to implement and comply accordingly. But if you empower them by training them, so it could be, yes, the biosafety officer could have been trained at advanced level, but now the biosafety officer should be able to train the rest of the staffs on biosafety and biosecurity, like now what we are doing in this webinar. So that should also apply in real world practice. Yes. And the risk assessment is done with the lab management as well as the biosafety officer. And you don't just do this by assuming. It means you have to do a proper evaluation of your system by engaging your staff, because they're the one who deal with the specimen on day-to-day basis. And by that, they'll be able to give you a clear view on how the system works. Yes.
Thank you, Francis. I think we've come to the end, and maybe I can take this back to you for your closing remarks.
Thank you so much, Fred. I'm sorry I was not able to log in by 3. I had a network hitch that was resolved. I think by 3:05 we were able to start. So we are looking forward to doing more topics maybe on biosafety and biosecurity, and I'll see what to do next around this area. But it is a gray area that we need to take very seriously, because every single sample that you have in the lab, if lands in the wrong place, like we have given an example, you are having pathological waste being disposed on landfill, you can imagine how you will be exposing so many people to the infectious agent. This could be easily washed to the river now, so things like that. So we really need to be keen on who handles our waste. That's a very important key, because especially us in Nairobi, you know, the good thing with the county hospitals or the hospitals, the public hospitals, especially the ones located in our counties, in the villages, they have got their own incinerator, or rather incinerator is not far. So they do treat their waste themselves. That is more safer. But here, where you are just going to give contract to anyone, you really need to vet the contractors and ensure they have got the proper infrastructure to handle your pathological waste that you are releasing to them. Yes, and also at least there should be a certificate that that company is accredited by the relevant bodies. Yeah, and the staff who are handling this waste are as well sensitized, and they are trained, because you cannot handle general waste the same way as the pathological waste. Yeah, thank you so much. Till next time, have a great evening.
Thank you, Francis, for the very insightful presentation, and also want to thank the participants who found time to join. I think it was quite a wonderful and very elaborative presentation, and I think we've just come to the tail end of today's session. And as Sad said when we were beginning, we shall be able to process your certificates and send them within 24 hours. So just check either your email, and also you can also check your junk folder or the spam box. Otherwise, it was such a great honor to have all of you. And until next time,